again and welcome to Excellence Icon. We'll be discussing now the lipids. Okay, so lipids are a mixed group of hydrophobic compounds, hydrophobic water heating compounds composed of the elements carbon, hydrogen, and oxygen. They contain fats and oils. Fats are solid at room temperature, whereas oils are liquid. This will be also discussing in the video later. The lipids so the complex structure of lipids are triglycerides. We'll be discussing it in this video. We have already discussed the phospholipid and the waxes, which is not a part of the syllabus. And the simple structures of lipids are steroids and terpenes. The steroids such as cholesterol, cholesterol, not estrogen. Estrogen is the hormone. Okay, the steroids are such as cholesterol, etc. Triglyceride is most important for you to understand. The structure must be on your fingertips. Okay. Triglycerides are commonly called fats or oils. They are made of glycerol and fatty acids. Glycerol is a small three carbon molecule with three hydroxyl group. Okay. This is the glycerol in front of you. You can see here. This is the glycerol. That three carbon, each carbon contains three not each carbon contains, yes, three carbon contains one hydroxyl group. Okay, so from the previous slide video we are starting with this. Fatty acids are long molecules with a polar hydrophilic end and a non-polar hydrophobic tail. So the hydrophilic head is visible to you here, yes, and the hydrophobic is also visible to you. The hydrocarbon chain can be from 14 to 22 CH2 units long. The hydrocarbon chain is sometimes called as an R group. R group. Okay, one misconcept for every candidate is that they just think so that the R group is the side chain for which we represent the side chain. It is not only just that this might be a chain. There might be only hydrogen, and this R groups. Remember one thing, that this R groups gives the variety. Okay, so this is the basic concept of the R group. Okay, it gives variety. All right, variety to the molecules. Yes. Okay, so the general formula of fatty acids can be written as R and COOH. Obviously, it is a carboxyl group. Okay, so now in your lipids, you can see here the hydrocarbon chains where the methyl group is present and the carboxyl group is also present. In the methyl group, we have already discussed in the organic chemistry. Okay, if there are no double carbon bond in hydrocarbon, then it is saturated fatty acids such as saturated with hydrogen. All right, these fatty acids straight chains and have a high melting point because they are saturated. As you can see that because they are saturated, they are fats, usually fats. Fats is a solid at room temperature and has a high melting point. Because it have a high melting point, it has a lot of melting point at the room temperature. It is solid. So in our body, the temperature is low than the room temperature sometimes. Yes, we can say it requires a lot of heat energy to break down. So fats are very harmful for us. Fats are very harmful for the human body in a large quantity. So obviously, how will be it break? Okay, and because of this, many times there must be heart attack or the coronary heart disease in the artery. Coronary artery, as we have already discussed the disease in the coronary artery. Yes, we have already discussed in the video that how does it appears due to the fat molecules which are accumulated veins in the arteries, and that causes the artery to stop the blood supplying, and that could be a rare case. All right, if there are double carbon bonds in the hydrocarbon chain, then it is unsaturated fatty acids such as unsaturated with hydrocarbons. Saturated with hydrocarbons, unsaturated with hydrocarbons. That's not understandable. Means that's not understandable to me. Means what you're trying to say. Okay, let's see. These fatty acids form band chains and have a low melting point. Remember this word band chains. We are coming back to that. Fatty acids with more than one double bond are called polyunsaturated fatty acids. That's not important for you to remember, okay? Now, from the structure, until and unless we does not see the structure, it is impossible for us to recognize that which is having a straight chain, which is having the branches, which is helical in the structure, and also that where the double bond present. Okay, in the saturated fatty acids, you can see that there is a straight carbon chain with no bending. Okay, you can see here in the saturated fatty acids there is no double carbon bond. All right, and every carbon has got the perfect amount of hydrogen. While in the unsaturated, you can see that there are two carbon which are not having the which are not having the hydrogen molecule attached to them due to the double bond. Due to the double bond, there might be possible chances that this is unsaturated with hydrogen. They requires hydrogen. Okay, they are unsaturated. Plus, they are not only just a straight chain. You can see that it forms a band chain, and this band shows you that this is unsaturated. Sometimes they give you the simple line, a straight line with nothing written, or sometimes they give you OH. Sometimes, but it's when it's straight line, you're required to recognize it's saturated. When it is straight but having a band on it, it means that this is not a straight line. Yes, and it is unsaturated fatty acids. Okay, once molecule of glycerol joins together with three fatty acids molecules to form a triglyceride molecule in another condensation polymerization reaction. Triglyceride are insoluble in water. They are used for storage. And one more thing is that more than glucose, if anything provide us the energy is the triglyceride are the lipids. Okay. One more thing also triglyceride. You eat your food which is cooking inside the oil. You're using the oil for your car batteries. You're using the oil for cleaning the window or for any purpose. These all of the oils or these all of the fats are the triglyceride. All right. Triglyceride contains okay. Carbohydrate can be mobilized more quickly, and glycogen is stored in muscles and liver for okay. Triglycerides contain a saturated fatty acids have a high melting point and tend to be found in warm-blooded animals. Okay, at room temperature they are solids, such as butter. Triglyceride contains unsaturated fatty acids, have a low melting point, and tend to be found in cold-blooded animals and plants. At room temperature, they are liquid, such as fish oil, vegetables. All right. We'll be discussing the phospholipid as we have already discussed this. It has a hydrophilic head and hydrophobic tails. Phospholipid have a similar structure to triglyceride but with a phosphate group. Okay, a phosphate group, you know, P. Okay, this is the determination for phosphate. Phospholipids have a polar hydrophilic head with the negatively charged phosphate group and two nonpolar. Okay, that's for we have already understand. Yeah, moving towards the structure. And mixed with water, phospholipid form droplet spheres with the hydrophilic head facing the water and the hydrophobic tails facing each other. And this is called micelle. Alright? This is structure. Name of this structure, okay? And here, alternatively, they may form a double layered, this is called a double layer bilayer, okay? Phospholipid bilayer. This is called the phospholipid bilayer. This traps a compartment of water in the middle separated from the external water by the hydrophobic sphere. This naturally occurring structure is called a liposome and is similar to the membrane surrounding a cell. Waxes. Waxes are formed from fatty acids and long-chain alcohols. They are commonly found wherever waterproofing is needed, such as in leaf cuticles, insects, exoskeletons, birds, feathers, and mammals fur. Steroids such as are steroids are hydrophobic molecules found mainly in animals. They are the cholesterol, bile salts, steroid hormones. Okay, that I was correct. Okay, steroid hormones such as testosterone, estrogen, progesterone, and cortisol. Vitamin D, which aids calcium positive 2 uptake by bones. Okay, and we're moving towards the protein. So, protein are the most complex and most diverse group of the biological compounds. They have an astonishing range of the different functions as this is listed below, and you can just pause the video and just have a quick review on that. Its structure, enzyme, transport, pumps. Okay, this was we have discussed in the chapter number 4. Motor, hormone, receptor, antibody storage, blood clotting such as fibrin and thrombin, lubrication, toxins, antifreeze, and many more. Okay. Proteins are made of amino acids. Amino acids or amino acids are made up of 5 elements. And the 5 elements you can see that there are the carbon, hydrogen, oxygen, nitrogen, and sulfur. The general structure of an amino acid molecule is shown on the right, or what we can say is below. There is a central carbon atom called the alpha carbon with 4 different chemical groups. This is what the determination for the protein, which I love the most for you guys to just remember. The from the middle of the carbon you can see what is present here: amine group, carboxylic group, the R group, which is the side, okay, which gives the variety, and the hydrogen atom. A single hydrogen atom is present. This is the determination for the protein. All right. Now answer me from this structure. That's where the amine group is visible to you, and the carboxyl group is visible to you. R group, including the hydrogen atom. Just tell me that why the hydrogen is not stored in your body. Okay, before you are taking enough time, let me just tell you the answer. Okay, so due to the amine group, or in other words, due to ammonia. Alright, when the protein is breakdown, ammonia is formed. So the part of amine group is removed in the urine or excreted from the body. Alright, and the other part of protein is stored or not stored, is used by the body. All right, that is why protein is not stored, because it is producing ammonia, and ammonia is a toxic gas for human beings, most dangerous. Okay, amino acids are called because they have both amino acids and acid group, which have opposite. Okay, amino groups and acid groups, sorry, which have opposite charges. At neutral pH found in most living organisms, the groups are ionized as shown above. So there is a positive charge at one end of the molecule and a negative charge at the other end. The overall net charge on the molecule is therefore zero. A molecule like this with both positive and negative charges is called, okay, this is not important for you candidate, Zwitterion. Okay, that's not important for you. Okay, and this is pH alkali. No, 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 that's not important for you. You're just required to just acknowledge the protein, the structure of protein, and the uses. Okay, now this is not important for you. Okay, okay. The 20 amino acids. There are 20 amino acids. Okay, and why they are different? So they are different due to the R groups, which provides the variety to the to the protein molecule. Okay, for interest only, no knowledge requires. Okay. Okay. That's not important for you. And the polypeptides are what we have been discussed. Polypeptides. Okay, polypeptide means you're required to just focus first on the peptide bond. First, focus on the peptide bond. In the when the two glucose molecules combine together, it is alpha or beta. A glycosidic bond was formed when two ammonia molecules. Sorry, not ammonia. Oh my god. When two protein molecules combine together, they form a peptide bond. And see how they are forming. The carboxyl group of one protein is reacting with another protein's amine group. From the amine group, two hydrogen are taken, and the double oxygen carbon bond, the oxygen is taken from the carboxyl group. All right. As you can see that this is visible to you candidates, I hope, including the hydrogen, yes, including the hydrogen of carboxyl group from the hydroxyl group. Okay, so from this diagram you can see that how they appearing that the carboxyl group, they are the hydrogen and one double bonded oxygen is taken. Not double bonded oxygen is taken. Yeah, the diagram here show you is not correct. Yes, due to the reason that from the protein molecule, the carboxyl groups, hydroxyl group is also taken, and it is the only which is taken, not the double bonded oxygen. As you can see from your diagram in the product, that they are the double carbon and oxygen bonded. Okay, and the CN with the carbon and nitrogen is the peptide bond. So where does the water molecule comes from? The one hydrogen of amine group and hydroxyl group of carboxylic group. All right. Yes, yes, yes. When two amino acids join together, a dipeptide bond is formed. Okay. Three amino acids form a tripeptide. That's not important for you. Many amino acids form a polypeptide. Obviously, when there is a more polypeptide, so it is called as where there is a peptide bond between two amino acids. There must be more amino acids, more peptide bonds. More means poly. Yes, polypeptide means more peptide bonds. Means there are amino acids. In a polypeptide, there is end with a free amino or amine group NH2, NH3 in solution group NH3. Okay, called the N terminus. Okay, that's not important for you. In the structure, which is very important, polysaccharides are just a string of amino acids, but they fold up to form the complex and well-defined three dimensions structures of working protein. To help to understand the protein structure, it is breakdown into four levels. All right, from the number one, primary structure, number two, secondary structure, number three, the tertiary structure, and number four, the quaternary structure. So from the primary structure, this is just a sequence of amino acids in the polypeptide chain. That's very simple. As a primary, it's a sequence of amino acids in a polypeptide chain, so it's not really a structure at all. However, the primary structure does not determine the rest of the protein structure. Finding the primary structure of a protein sequencing, and the first protein to be sequenced was the protein hormone insulin. Okay, in the secondary structure, this is the most basic level of protein folding and consists of few basic motifs that are found in our protein. The secondary structure is held together by hydrogen bonds between the carboxyl group and the amino group in the polypeptide backbone. The two secondary structures are the alpha helix and the beta sheet. Alpha helix and the beta sheet. The helix means helical shape, okay, where the polypeptide bond is shown to you and the hydrogen bond is also shown to you. Okay, the alpha helix. It means that the polypeptide chain is wound round to form a helix. It is held together by hydrogen bonds running parallel with the long helical axis. There are also so many hydrogen bonds that is a very still and strong structure. Helical helixes are common structures throughout biology. Yes. And the beta, and in the beta, this is the beta sheet. The polypeptide chain zigzag back and forward forming a sheet. Once again, it is held together by hydrogen bonds. All right. So this is called the beta sheet. The structure is very important. And the tertiary structure, it is a three-dimensional structure formed by the folding up of the whole polypeptide chain. Okay, hydrogen bonds which are weak, obviously, which are present here. There are the three kind of bonds involved: hydrogen bonds and the sulfur bridges. There were the covalent SS bonds. that is making the tertiary structure of the protein. And the ionic bonds between R groups with positive and or negative partial charges, which are quite strong, obviously. Covalent SS or sulfur-sulfur bonds between two cysteine amino acids, okay, which are strong again. Remember these three bonds are present here, okay? And quaternary, which is shown to you. So they just show you the quaternary structure. That's cool, okay? This structure is found only in proteins containing more than one polypeptide chain and simply means how the different polypeptide chains are arranged together. The individual polypeptide chains are usually globular but can arrange themselves into a variety of quaternary shapes, such as hemoglobin. The oxygen carrying protein in red blood cells consists of four globular subunits arranged into a tetrahedral pyramid structures. Each subunit contains one iron atom. Heme basically contains one iron atom and can bind one molecule of oxygen. Yes. One of the most important, okay, that's what you can just pause the video and see. One of the most important thing is what is the difference between the globular and the fibrous protein. Oh, sorry, not protein, the structure. This is the structure of globular protein and this is of the fibrous protein as is visible to you. Okay. And now from here, I do not know that is this part of your syllabus. Globular protein is part of your syllabus and the fibrous also. But this explanation I does not know. You can pause the video and just see. It's a alpha helix from and alpha helix from side. Okay, random loop and band. Yes, three strands of sheet. Okay. The diagram shows a molecule of the enzyme. Okay, so this is the diagram for the enzyme which compromises a single polypeptide chain. It's a globular protein, okay, and that is the okay. Okay, it means that this single line of the globular protein inside three strands of beta sheet. So this is called the globular protein, okay, and a single line is called the fibrous. I hope so that this is one of the most important for you to acknowledge. So this is one, and that's all. And another thing which is the biochemical tests. Okay, so this is we have already discussed it. For that, I'll just making you understand. You can just pause the video and see here. Okay, and what is that? Protein, carbohydrate, lipids, and that's the difference. You can just pause the video and just see it has a quick revision to you candidates. Okay, and the carbohydrates keynotes. Okay, keynotes are here, and the monosaccharides. Okay, disaccharides. These are the one of the most which I've been discussing at the end. Wow. And the polysaccharides. Okay, so that's all. That's all. I hope so that you have understood about everything, every of structure, carbohydrate, and lipids. If you are still confused, do not worry. Just comment me below. I'm just waiting for the answer. And this is the quick review. You can take the screenshot or can write it as a note. Okay. And I prefer candidates to just make the notes by in their own language so that it could be more visible to understand. Okay, and you will be able to just solve the paper. Do solve past papers also. Okay, past papers are for most important for you, and there you can also acknowledge that how the structures are given to you. Okay, so till that's it here. I hope so that you understand. If you does not understand, comment below whatever is confusing you. Till that is here, take care, goodbye.